Allow me to introduce you to the upgraded Deepchain platform. We have here the models page. We can get access to different models. For example, we've got the DNA models here, the segment NT that we've been discussing. Here we have the specific information about its datasets and different training parameters. We've also got our protein models that have been introduced, like ProtBFN and AtBFN. And finally, we can jump into these fine-tuning capabilities we've been discussing. In particular, you can start a new run by clicking on this button. You can now select which model you'd like to use for fine-tuning. We're going to be adding a name here, AI Day, for this run. We'll be selecting the downstream task for gene expression. And now we'll be uploading our fine-tuning data. So in particular, for fine-tuning, you need a file that's your file that contains your sequences. And then you have your training labels. In addition, you can also upload your validation data, so both your sequences and your labels. And then down here, you've got some parameters that you can fine-tune. And this is actually the part where scientists can experiment and test different parameters, different type of data. So biologists are really the experts in the data, while we can handle the compute and the orchestration on our side. And so here is where they can actually start experimenting. So now we're going to start the fine-tuning run. The data has been uploaded. And now we can go on to the runs and actually keep track of this processing run here that we just started on AI Day. So what we see here is the different parameters, and the plot is being updated live with the results that are being processed. But because usually fine-tuning takes quite a few hours to be able to achieve good results, we're going to use a run that I started actually earlier this morning that succeeded, and also a model for AI Day. And so here we see how, so the plots have been completed, and we're able to use this model now for inference, to be able to show how it works in practice. So now we just added the model to our list, so it's the one here. We're going to be jumping onto the CLI for you to see how we can run these models. So here I'm going to start by typing Deepchain models to see the list. So we just got the list of Deepchain models. In particular, this is the one here we'll be using today. And then to run our models, we're going to be typing Deepchain run. Then select the model that we just moved to our list. Then referencing our sequences that we want to use and our output file that we will be sending the predictions to. And we're going to ask it to wait so we can see what's happening. So now what we did is just send some sequences to our systems, and what's happening in the background is because we're using a fine-tuned model for gene expression, we're going to be seeing for each sequence that we've sent through, we're going to be seeing a few values predicted for gene expression in the different tissues. So we just got back the results. There's a snippet of those results where we can see the gene ID associated with these different values of gene expression. And now we will be using, so this is where we can see the full results. So we'll be using this file to be able to evaluate the results. We have a function here on Deepchain where you can evaluate, so you can type Deepchain evaluate, yes, and then we can reference our results that were just computed, and a list of labels. Sequences underscore labels is where our labels are. And so now our results pop up here. We see the performance improvement of our fine-tuned model that we've just showcased compared to a baseline. And so you can see, as you can see, if you really play around with those parameters and your data sets, you're able to achieve improved results with our models and customized results to your application.
My name is Anu Pretorius. I'm a research scientist at InstaDeep, and I'm delighted to introduce to you today our AI agent called Laila. Laila is deeply integrated into all aspects of the Deepchain platform, including running models, performing analyses, calling internal as well as external tools, and much, much more. So to showcase some of Laila's capabilities, we're going to run through a hypothetical scenario of a user who's new to the Deepchain platform and interested in analyzing DNA. So I'm just going to clear here at the side. Let's clear this, and we can go to Laila. So being new to the platform, we might begin by simply asking Laila, which models can I use for DNA? Laila then knows to call the internal API associated with the Deepchain platform and give us a list of available models that a user could be using to analyze DNA. We see segment-NT, a multi-species version of segment-NT, and a whole host of other fine-tuned models based on the nucleotide transformer series built by InstaDeep. Now, being new, this user might not be familiar with these models and might be interested in knowing what they can actually do. So we can ask Laila, what is segment-NT used for? Again, Laila calls the internal API, but now fetching information specific to segment-NT, giving us the reply saying segment-NT is used for detailed genomic analysis, offering single nucleotide resolution predictions for various genomic elements. So this might sound interesting to us. So we can go one step further and try to do some analyses. So if I just pull up a DNA sequence file here, I can upload this to the platform, and you'll see this under the attached files being shown over here. Now I can tell Laila that I have uploaded a sequence and tag it using the @ symbol. Once this is done, I can then ask Laila, can you please segment it for me? Laila now knows that it can call the custom segment-NT model and give as input the DNA sequence that we've just uploaded. It provides us a segmentation text output saying that at specific indices within the sequence, we can find exons, introns, splice donors, splice acceptors, as well as UTR elements. But not only this, this gives a scientist a quick sort of overview and a feel for what's going on, but they might want to actually build a much more custom pipeline in Python, some sort of larger scale batched workflow. And as Julia showed, we have the CLI for this that can really be useful. And to get started, Laila provides us with a command we can actually directly copy and paste to run this analysis through the CLI. So I'm just going to pull this across. And here, if we just quickly look, we have this DNA file here that we uploaded. And we can simply paste this, knowing that this is exactly the DNA file we want to analyze with the output being specified. When we run this, the CLI goes and fetches the job, does the analysis by calling the model, and provides us with the output. So this takes just a few seconds. And here we can actually take a quick look at this output file. And what we can see is it has a whole host of probabilities of certain regulatory elements being in certain places. And even though this is not nice to look at through the terminal, but in a Python workflow, you can very much use this easier for further analysis. But we can now go back to Laila and simply ask, if we just want to, again, have a sort of overview of what's going on, to visualize the results for us. Can you please plot it for me? Laila knows it has an associated plotting script with segment-NT and can run this to give us an interactive plot of what's going on. So on the y-axis here, we see certain regulatory elements, and on the x-axis is the nucleotide positions in the sequence. And what we're seeing here is probabilities of certain elements being present at certain positions. And we can actually zoom into specific regions and see that here, for example, the nucleotide A in position 1490 has a probability of 92% of being associated with a tissue invariant promoter. And this can be a nice tool to really improve the productivity of biologists to quickly get going with the Deepchain platform.